The overall goal of this procedure is to assess the olfactory function in mice. This is accomplished by first putting the mice on food restriction or individual housing. The second step is to prepare the olfactory stimuli, setting up the testing room, and habituate the mice to the testing area so that the tests can be performed. Next, conduct the testing procedures on the mice and analyze the data that was recorded on the video. Ultimately, these tests can be used to measure the olfactory abilities in genetic mouse models of neurodegenerative disease and in clinical drug studies to test potential therapies.
Although olfactory deficits are well documented in neurodegenerative diseases, olfaction is not routinely measured in many disease models. Therefore, our goal was to assemble a battery of tests to measure multiple aspects of olfaction in mice that can be easily implemented into an experiment.
These tests can help answer key questions related to the relationship between neurodegenerative disease pathology and olfaction, as well as to help identify the therapeutic potential of novel compounds.
To begin this procedure, give the mouse 1 to 2 pellets to be used during the test daily. Then, at least 2 days before testing, record the weight of the mouse and then food restrict it to 90% of its body weight. On all the test days, habituate the mouse for 1 hour prior to testing by placing the home cage in the testing room with just a filter top lid. During habituation, fill a clean cage with clean bedding to 3 centimeters high and make sure that the bedding is evenly distributed throughout the cage.